Imagina poder construir
En este video te voy a enseñar cómo agregar el chat de Twitch a tu stream y solamente dando un clic. Así que si quieres saber cómo hacerlo...
Bueno amigos, hoy vamos a ver cómo poner el chat de YouTube para tus dudas. Vamos a irnos en cómo se contesta aquí en el OBS y en la clic en aceptar y aquí vamos a buscar y cuando nosotros esto ya tenemos aquí es que tiene, vamos a buscar, comparte lo que tan flotante y lo que yo voy a hacer es redimensionarla ancho hasta donde ustedes quieran, ustedes lo pueden hacer, vuelvo a llevar. Y bien, ahora para quitarle esta parte acabamos de añadir para que ustedes miren a buscar el modo de fusión. Le vamos a dar en añadir para que ustedes miren el efecto. Entonces miren cómo el chat se vuelve más transparente o se vuelve, ustedes miren y vamos a buscar el modo de fusión. Le vamos a dar en añadir para que ustedes miren el efecto. Entonces miren cómo el chat se vuelve más transparente o se vuelve transparente y se vuelve un poco más agradable para la vista de los espectadores o en la pantalla se ve mucho más agradable. Ustedes quieren experimentar, pues hacen lo mismo, clic derecho en la fuente, modos de fusión, le vamos a dar
¿Qué tal, amigos? Espero que tengan los streams. Algunos aspectos. Nos vamos a dirigir a OBS. Les voy a mostrar aquí OBS, pero mi OBS, tus fuentes. Vamos a darle en más aquí pegar asiento. Ya te dice cuenta que aparece, lo tenemos, ¿ok? Listo. Rectángulo, vertical, algo. Voy a mi canal y aquí está. Listo. Bueno, y así de fácil. Aquí ya empieza a aparecer esta cuenta en mi OBS, aquí en la parte de abajo, aquí más grandecito por acá, así y así. Bueno, yo aquí voy a empezar a comentar. Hola, hola. Vamos a empezar a transmitir algo súper rápido, simplemente para hacer la Streamlabs. Ancho 800, alto vamos a darle en 1080 porque yo estoy transmitiendo en 1920 por 1080 y así lo tenemos, ¿ok? ¿Listo? ¿Ya? Ya te diste cuenta que apareció algo aquí como un rectángulo vertical, ¿sí? Vamos a empezar a transmitir. Vamos a empezar a transmitir algo súper rápido, simplemente para hacer la prueba. Iniciar transmisión. Yo ya lo tengo configurado, ¿ok? Me voy a YouTube, voy a mi canal y aquí tengo el directo, ¿ok? Aquí está. Yo aquí voy a empezar a escribir. Voy a tratar de dividir la pantalla de mi OBS junto con mi transmisión. Aquí está. Perfecto. Un poquito más grandecito por acá. Así. Y así. Bueno, yo aquí voy a empezar a comentar. Hola, hola. 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 Por acá.
अस्सलाम वालेकुम।
वालेकुम सलाम।
और भाई ठीक हो?
जी
अच्छा
ठीक।
ਜਿਹੜੇ ਉਹ ਮੈਨੂੰ ਸੈਂਡ ਕੀਤੇ ਨੇ ਸਿਰਫ ਇਹਨਾਂ ਤੇ ਕਲਰ ਕਰਾਉਣਾ ਹੈ ਕਿ ਪੂਰੇ ਕਮਰੇ 'ਚ ਕਰਾਉਣਾ ਹੈ ਕਿਉਂਕਿ ਉੱਪਰੋਂ ਰੰਗ ਲੈ ਕੇ ਬੰਦੇ ਦੇ ਆਉਣਾ ਹੈ ਨਾ
नहीं बस वही है।
ਸਿਰਫ ਇਹ ਦੋ ਨੇ
जी
ਤੇ ਇੱਥੇ ਉਹ ਜੋ ਤੁਹਾਨੂੰ ਛਿੱਲ ਕੇ ਦੋ ਜਿਹੜੀ ਇਹ ਸਪਰੇਅ ਵਾਲਾ ਮਿਲਦਾ ਪਤਾ ਜੀ ਇੱਕ ਕਲਰ ਉਹ ਮਾਰਦੇ ਤੇ ਨੋ ਪ੍ਰੋਬਲਮ ਹੋਰ ਕੋਈ ਸਫਾਈ ਸਫੂਈ ਵੀ ਕਰਨ ਵਾਲੀ ਹੈ ਕਿ ਸਿਰਫ ਇਹ ਦੋ ਚੀਜ਼ਾਂ ਨੇ
बस जाइए थोड़ी, हाँ बस जाइए सफाई थोड़ी कर लेते हैं बस।
ਚਲੋ ਵੀ ਕਰ ਦੇਦਾ ਜੇ ਤੇ ਨਾ ਉਹ ਸਵੇਰ ਇਨਸ਼ਾਅੱਲਾ ਅੱਲਾ ਖੈਰ ਕਿ ਜੇ ਤੁਹਾਡਾ ਰਾਬਤਾ ਕਰ ਲੇਦਾ ਤੇ ਕਿਸ ਟਾਈਮ ਤੁਸੀਂ ਫ੍ਰੀ ਹੁੰਦੇ ਹੋ ਸੈਕੰਡ ਟਾਈਮ
नहीं मैं तो कल जा रहा हूँ।
कल किधर जाना है
मैं माजोर्का जाना है कल।
तो फिर वापसी कब है आपकी
8 को है, 7 को।
तो फिर सही है फिर ऐसे प्रोग्राम बना लेते हैं ना
नहीं नहीं
ये
आगे
रात को ना
आगे नहीं आ सकते आज, आज नहीं आ सकते।
आज नहीं आ सकते क्योंकि उनको एक बार पहले बोला था आने के लिए फिर आपके पापा को याद नहीं रहा आज फिर उन्होंने मुझे फोन किया और मैंने उसको किया आज वो कहीं काम पे आज मुझे दिन को उन्होंने फोन किया आपके पापा ने ना तो वो कहीं काम पे गया हुआ है तो वो कह रहा आज मैं लेट हो जाऊंगा तो मैं सुबह इंशाल्लाह ना जो वो
कल
अपना
किस टाइम आ सकता है?
तो इस तरह करते हैं कि 8 को आपको ना वो बेहतरीन कलर कर देगा सारा सही है
ठीक है।
आगे आपका नंबर नहीं था मेरे पास 2-3 बार मैंने आपके पापा को कॉल की उन्होंने अटेंड नहीं की जब वो मुझे कॉल करता रहा तो उसके साथ प्रोग्राम बना लेते हैं जब आप उधर उतरेंगे ना आप सारा रूम भी सफाई कर देगा और कलर भी कर देगा सारा ठीक कर देगा सही है
ठीक है।
जी सही अल्लाह हाफिज
हाँ। सात को School.com, school.com, school.
Este es Jorge.
Hello Ledger explains the process for accepting. We hook it up for SMSs. We plug it into our WhatsApp. We can do SMSs. We just spoke about those call experiences. And then, yeah, so like we discussed before, instead of us having to write Twilio, at least do SMS and voice so that you can have a consistent experience for your customers. So yeah, leave everything highlighted. I reckon it's fine. And the second thing is, we've noted, so just, yeah, your business.
With the procedures. So of course, for Twilio, we have a 4-step process. First, we'll have to create a Twilio account. Then we'll have to buy a number, and then we'll have to create a sender. So creating a WhatsApp with a number that you've purchased, and you need to allow voice calls. Now, when you have done that, it will be reflected in a few minutes. For my case, it took me almost 5 to 10 minutes. So after 5 to 10 minutes, your WhatsApp account will have that call icon. Now that you've done that, your Twilio as well as your WhatsApp number is interconnected. So this particular part is done for us. Now we have already
En este video vamos a describir las mutaciones del ADN. Muchos de nosotros habremos escuchado alguna vez que muchas de las enfermedades están asociadas a mutaciones genéticas o a mutaciones del ADN. Pero muchos de nosotros no sabemos por qué se dan esas mutaciones o cómo es que se producen o qué incidencia tienen sobre las proteínas que se generan. Acá nos vamos a tomar unos minutos para conocer cuáles son los tipos de mutaciones más comunes del ADN. Vamos a ver los tipos de mutaciones respecto de las causas de esas mutaciones. Vamos a ver primero la más común de todas, que se llama la deleción. En el caso de la deleción, lo que vamos a ver es, por ejemplo, en este esquema que tenemos acá, que falta, vamos a hacerlo más chiquito, que falta, por ejemplo, el nucleótido adenina. Lo vamos a tachar así. La adenina desaparece y entonces ya no tenemos adenina en el ADN. Eso es la falta o la ausencia de uno o más nucleótidos. Así se define.
Con solo 20.000 ahorrados y este documento sabrás cómo comprar y alquilar un piso que te genere.
Por otro lado, tenemos a la inserción, que sería el opuesto a la deleción, donde se adiciona o se incluye uno o más nucleótidos, y lo vamos a ver, por ejemplo, si pudiéramos incorporar en este caso una timina en el ADN, y todas estas deleciones o inserciones van a causar que cuando este ADN sea transcripto a la molécula de ARN y después pase a la proteína en el proceso de traducción, va a generar una proteína que es diferente. Eso ocurre con las deleciones y las inserciones, al igual que ocurre con otro tipo de mutaciones que se conocen como las sustituciones. En el caso de las sustituciones, se van a agregar se van a cambiar, no se van a agregar, se van a cambiar los nucleótidos. ¿Cómo es que se cambian esos nucleótidos? Es, por ejemplo, en este caso, se borraría la adenina, pero en su lugar se colocaría una citosina. Es un cambio de nucleótido. Se va la citosina y en su lugar aparece una timina. Eso también es bastante común de encontrar y genera proteínas que son diferentes a las que estaban en el código genético. Y otra de las últimas que vamos a ver, que son las más comunes dentro de los tipos de mutaciones, es la conocida como la translocación. La translocación. En este caso, y es muy común que se dé durante la recombinación, vamos a ver que fragmentos del ADN se intercambian. Vamos a tener, por ejemplo, en los aminoácidos de la proteína final. ¿Y cómo es que se puede dar eso? Si pudiéramos hacer un ejemplo de la cadena de ARN, la cadena de ARN mensajero que se transcribió desde el ADN por el proceso de transcripción, vamos a dibujar, por ejemplo, una cadena de ARN. ¿Se acuerdan que ya acá no hay timina, sino que hay uracilos? Entonces tenemos adenina, adenina, uracilo, 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 adenina, citosina, guanina, guanina, citosina, y esto entonces es un ARN mensajero. Cuando en el ARN mensajero, si se acuerdan, teníamos los codones. El codón era el triplete o las tres bases que formaban este codón, que es el marco de lectura que se le va a dar para el proceso de traducción de proteínas. Entonces acá tenemos un codón, otro y otro. Si en este caso vamos a prestar atención al codón de la triple uracilo. Este codón codifica para un aminoácido conocido como fenilalanina. Pero si tuviéramos una mutación silenciosa, lo que se ve es que se cambia un aminoácido por otro. Un aminoácido, vamos a ponerle aminoácido 1, se cambia por un aminoácido 2. Esto se ve claramente, por ejemplo, si existe una mutación aquí y se cambia por otra letra, otra base, esto sí genera un cambio en el codón y va a codificar para otro aminoácido activo, genera producción para la función de inserciones o adiciones. Lo que se ve es que se adiciona un nucleótido, por ejemplo, esto se ve mucho en las adiciones o en las deleciones. En las inserciones o adiciones, que es lo mismo, inserción o adición, o en las deleciones. Cuando un nucleótido se incorpora o falta en el ADN o en el ARN, transmitido al ARN, lo que sucede es que si yo tenía este marco de lectura, iba leyendo codón 1, codón.
The mRNA. Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish, or a cat, or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule, consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism. Thanks for watching, guys. Subscribe to my channel for more.
En materias como biología, los detalles de un fenómeno o proceso se pueden comprender mejor si se exponen de manera lógica, transitando oportuno, coherente. Los detalles al respecto abruman. Son como los bloques básicos con los que se construye un ser vivo, como el gato Wally, o como tú o como yo. La copia o transcripción del ADN en ARN se realiza en el núcleo celular, en el núcleo con membrana de la célula eucariota y en el núcleo sin membrana de la célula procariota. Pero la traducción o síntesis de proteínas sucede en una estructura celular llamada ribosoma. Aquí apreciamos un par de ribosomas en el citoplasma. Parecido quizá a una bellota, el ribosoma se compone de 2 partes, una menor y otra mayor. Aquí hay un montón de ribosomas en la célula procariota.
Nos preguntamos hace milenios qué es la vida.
El ADN se aloja en el núcleo y se organiza en cromosomas como el de esta célula eucariota. Por razones que no voy a enumerar aquí, el ADN del núcleo no puede pasar directamente al citoplasma. Pero los fragmentos de ADN transcritos en forma de ARN sí pueden salir del núcleo y llevar la información o mensaje al citoplasma. Está muy claro por qué a este ácido ribonucleico se le llama ARN mensajero o ARNM. A diferencia del ADN, que se compone de dos hebras, el ARN es una molécula de una sola hebra o cadena de nucleótidos. Recordemos que cada nucleótido de ADN está formado por un azúcar desoxirribosa, una base nitrogenada y un grupo fosfato. La desoxirribosa es un azúcar de 5 átomos de carbono. Los estoy señalando con el indicador láser. En química numeramos estos carbonos a partir del que se une a la base nitrogenada. ¿Ves? Tenemos 5. El quinto y tercer carbono son dos extremos importantes. A estos extremos se les llama 5' fosfato y 3' hidroxilo. He aquí el esquema de un nucleótido. Tiene sus extremos 5' fosfato y 3' hidroxilo. En estos extremos se encadenan otros nucleótidos para formar una hebra del ADN. De esta forma tenemos una hebra en sentido 5', 3'. Por su estructura molecular, la otra hebra de ADN forzosamente tendrá un sentido inverso. Las hebras del ADN son antiparalelas. Para copiar un polimerasa construye la R de cada base nitrogenada para tras ensambla de igual forma en una secuencia de bases nitrogenadas de aminoácidos. El mismo aminoácido y por eso se dice que a nitrogenadas o triplete se le llama codón. En la síntesis de proteínas, un codón se puede traducir en un aminoácido o en una señal de inicio o terminación del proceso de traducción. Varios tripletes o codones pueden codificar el mismo aminoácido y por eso se dice que hay codones sinónimos. La traducción se inicia con la unión del ARN mensajero al ribosoma a partir de un triplete o codón de iniciación que usualmente es adenina, uracilo y guanina y que también se traduce en el aminoácido metionina. Observa que el ribosoma abarca 2 codones del ARN mensajero que se van a acoplar a 2 ARN de transferencia. El ARN de transferencia es, digamos, como una pieza de rompecabezas. En un extremo coincide con un codón del ARN mensajero y en el otro lleva un aminoácido específico. Es así un transporte de aminoácidos. Cada aminoácido que transporta el ARN de transferencia se une o encadena con el siguiente mediante un enlace peptídico. Cuando eso ocurre, el ARN de transferencia que lo transportó queda libre y puede ser utilizado para transportar el mismo tipo de aminoácido en otra síntesis de proteínas. Observamos cómo el proceso continúa y se siguen encadenando o enlazando aminoácidos transportados por el ARN de transferencia. El ribosoma hace un recorrido por los codones del ARN mensajero en el sentido 5', 3'. El proceso de traducción termina hasta topar con un codón de finalización al que no le corresponde ningún aminoácido, es decir, no se traduce en ningún aminoácido, solo es una señal de fin. En este punto el ribosoma se separa de la...
Tengo un chat GPT ahora mismo abierto sin adulterar, nuevo, virgen. Y mira la tontería de idea de negocio que le doy y me la...
mRNA is very, very important for this translation process, right? Messenger RNA. Messenger RNA it has a very specific sequence of nucleotides, if you will, that are in these triplet forms. You see how this is there's 3 little lines there? That line is a nucleotide, that's a nucleotide, that's a nucleotide. So there's 3 nucleotides there. And we have a couple of these spanned along the length of this mRNA molecule, right? And the other thing you need to know is the orientation, right? So a little bit about the topology of the mRNA. On this end, I'm going to have a 5' cap. You guys remember that whole process we talked about in post-transcriptional modification? This is the 5' end. On this end, you have the 3' end. And we had on this end, you guys remember what happened here in the transcription process? We've added a poly A tail on that, right? So on the mRNA, you have a 5' end, a 3' end, and sequences of nucleotides within it. These sequences of nucleotides that are in these triplet forms along the mRNA are given a very special name that you need to remember. And these are called codons. So let's write that down. So these things, these triplets, let's put down triplets. Triplets of what? Triplets of one of those codons. You read them, right? Different types of possibilities. We'll code for it a little bit more in detail when we go through the phases of translation. Are a triplet, right? So 3 nucleotides that are complementary to the codons in mRNA for what amino acid? Methionine. So then this enzyme, phosphate group. This end over here, which has like a little copper loopy thing, is the 3' end. 3' end contains what? OH or phosphate group? Contains the hydroxyl group, or the OH group. But there's also a very specific sequence of nucleotides that are in this area of the 3' end which hold on to the amino acid. This is the amino acid holding domain. And this is containing CCA. So we'll put the 3' CCA domain or region on the tRNA. What is this portion? A little cup that holds on to what? What will bind in here? So here you have CCA. It'll bind onto the amino acid. In this case, was what? Fine. If the anticodon is UAC, which is complementary to AUG. Holy crap, we went through all that. All right, so the next thing I'm going to talk about is the characteristics of the genetic code. I don't want to go too long into this. Let's just breeze over really quick. But it's things that can be asked in your exam, so you should know it. When we talk about the genetic code, all the stuff we talked about, the codons, the anticodons, the mRNA, tRNA, all that good stuff. When we take the mRNA and we read it, right, from 5' end to 3' end, for the most part, there's a couple exceptions. You start at the 5' end and you go to the 3' end continuously. You don't like have any stops or anything like that. So in that way, when we talk about the genetic code, this translation process, understanding the genetic code, is what's referred to as commaless. So what does that mean? Here's a codon, right? I'm going to read this codon, utilizing the tRNA and the ribosomes, and I'm going to get an amino acid. Then I'm going to go to the next codon, read that, make amino acids. And I'll keep going down this way, going through each sequence of 3 nucleotides or a triplet. Now, what does it mean that it's commaless? Let's say that I read this codon, and there's a couple nucleotides in between this next codon that I want to read. I don't skip these nucleotides and go to the next codon, okay? So this thing does not happen. You don't go 3 nucleotides, read 3 nucleotides next, and then skip a couple process that does have commas in it, or you kind of skip a couple nucleotides, okay? The next thing that we need to know about the genetic code is that not only is it commaless, but it's non-overlapping. What does that mean? That means that when I read these, again, 5' to 3', I'm going to read it all the way down continuously. I'm going to read this codon, give an amino acid. Read this codon, give an amino acid. What I'm not going to do is read this codon, give an amino acid. But then, let's do a different color. Start here at the second nucleotide, read these 3, and give an amino acid. Let's do one more color. Start at this third nucleotide after, and read here, and give an amino acid. That does not happen in the translation process according to the genetic code. There is one exception, and again, that exception is that this overlapping process can occur in viruses. That is the only exception, okay? So when someone says, can you give me characteristics of the genetic code, you will say it is commaless, it occurs continuously from 5 to 3, and non-overlapping, continuous from 5 to 3. The only exceptions are viruses. That's it. The next thing that you need to know is a little bit more important than this gibberish up here, and that is that the genetic code is what's called redundant. So it's redundant, and the code challenge. Going forward, today, you do the same thing over here. Start at 5', 3' for the tRNA. Work your way down. First one has to be I. Next one will be what? A. The next one will be U. Let's explain what happens with all this. Do you notice the difference here? Remember I told you that the enzyme has to read that anticodon. It has to be complementary to the codons in the mRNA to pick the correct amino acid. Well, do you notice how all these are they all differ in that third position on their codon, and they differ on this kind of like first position in the anticodon? Here's how this happens. Inosine, okay, that I'm representing with is actually called inosine. Inosine is not talked about too often in the Watson and Crick model, in the DNA stuff, interactions, complementary stuff. Inosine is complementary to adenine. Inosine is complementary to uracil. Inosine is complementary to cytosine. So whenever you have something like this, where the third position is A, C, and U, on the tRNA, they can have an inosine that can be complementary to A, complementary to U, and complementary to C, and still give you the same amino acid, which would be what? I already told you this would be isoleucine, isoleucine, isoleucine. Okay? So that's called the wobble effect. You're probably like, why the heck do we do this? Why don't we just make it specific to each of these types of codons? Why don't we just make it UAU, UAG, UAA? Why don't we do that? The reason why is the wobble effect reduces the risk. It decreases the risk of mutations. So what do I mean by it can decrease the risk of mutation? And specifically, it can decrease acid will bind. Correct. Now, this arm was the one I really wanted you to focus on. We had the 3 loops. We'll briefly talk about these other 2 loops. Not super worried if you guys know, but here in this bottom loop, what do we have down here? On this bottom loop, you contain the anticodons. And the anticodons will be in a triplet form. And these triplets can be in the form of, again, containing nitrogenous bases like what? Adenine, adenine, guanine, uracil, and the cytosine. Okay? So again, this portion here will be what? This will be the anticodon portion. Okay? The last thing here is these 2 little arms or loops, and this little thing is kind of like sticking out the side. This portion here, near that 3' end, this is called the T arm. So what is this portion here called? Near the 3' end, this is called the T arm. The T arm, what you really need to know about this is that it tethers the tRNA to the ribosome. That's all I want you guys to know, is that it tethers the tRNA to the ribosome. So it kind of is one of the big things that allows for interaction between the tRNA and the ribosome. Okay, that's it. This other arm over here, this loop near the 5' end, this is called the D arm. And the D arm is what allows for the identification of the tRNA by the enzyme called tRNA, the amino acyl tRNA synthetase. So it allows for identification of the tRNA by what's called the, we'll have a basic thing, tRNA synthetase enzyme. Okay? So basic concept here, you have the 5' end, then you have the first arm, which is the D arm, allows for the identification of the tRNA by the tRNA synthetase. Anticodons on the bottom loop. T arm is near the 3' end, that allows for the tRNA to interact with the ribosome. 3' end has the CCA domain, which allows for it to interact with amino acids. This last thing here, I'm not really concerned if you guys truly know it, it's the invariable domain. It's the variable domain, it can change from tRNA to tRNA. Nothing too big to know about that portion. Okay, so the basic structure of the tRNA. The next thing I need you guys to know about is called charging. So this is really simple. It's basically talking about how do we get the amino acid to bind on to that 3' end. That's all it is. And it's really simple. Here I have an amino acid. Okay, so here's my amino acid. And let's use this example that we've continuously been using a lot. Let's say that we're going to start the translation process, and let's just pretend, for example, here's my mRNA. Okay, let's use this example that this is AUG. What would the anticodons be if we were to kind of write them in here? If it was AUG, it would be UAC. What is that AUG code for? Methionine. We've already said that multiple times, right? So let's say here's our example. This is our methionine. We'll just abbreviate it as MET. Okay. What we're going to do is, the first step we're going to do in this process is we're going to add an ATP molecule onto it. We're going to add an ATP molecule onto the methionine. So let's say that here, I use an ATP, and I add it into an acyl AMP molecule. Okay? Then, here's the next thing. We have this amino acyl AMP, and we have that 3' kind of amino acid domain with the CCA portion. Imagine we draw a big old enzyme here. So here's this enzyme. Okay? Here's this enzyme. This enzyme has in one end is holding the tRNA, right? So it's holding that tRNA molecule. So we're gonna draw a very generic structure of it. Here's gonna just be this process here, okay? So here's the generic structure, and we'll just kind of show that this is our 3' end right there. Okay? Just generic. It's holding in one pocket this tRNA molecule. And the other pocket, it's holding the amino acid with what bound to it? The AMP. Then what it does is, it basically just says, hey, let me make sure that this anticodon is appropriate. Is it appropriate to the mRNA codon that we need? Oh, it is, good. Clicks them together. And so it takes and adds that amino acid with the AMP onto the 3' CCA region. So let's draw a little cup. What was that little cup thing? The CCA portion. It'll add on, this reaction will occur, so we're gonna just fuse these two things together. And when we fuse these two things together, what do you get? You'll get this structure where you'll have the tRNA with a little cup, and what will be kind of sitting in that little pocket there, the amino acid. And what amino acid was this in this example? Methionine. In the process though, do you see AMP still bound to it? No. So what are we gonna do? We're gonna release the AMP during that process, okay? What you need to know is what the heck is this enzyme? This enzyme is called the amino acyl tRNA synthetase. I kind of quickly abbreviated it for you, like a shorthand version of it when we talked about the D arm. That's the enzyme I'm really referring to, is the amino acyl tRNA synthetase. And if you really want to remember what part of the DNA is keeping it kind of like identified, the D arm of the tRNA will allow for it to be identified. Okay? So, to recap really quickly, I want to take the amino acid, put it on the 3' end. What do I have to do? First thing, take the amino acid, add an ATP onto it. I'll pop off a pyrophosphate, so I'm truly only adding an AMP. That's called an amino acyl AMP. An amino acyl tRNA synthetase will come in, have two pockets. In one pocket, it'll hold the amino acyl AMP. In the other pocket, it'll bind the tRNA with no amino acid. It'll read, make sure that it's the proper anticodon that is complementary to the codon of mRNA. Click them together. When it clicks them together, it puts the amino acid on the 3' end and spits out the AMP. Now what do I have? A charged tRNA. So what is this thing here called? This is called a charged tRNA. Okay? That's the process. That's all I really want you to know out of this, okay? So let's now move on to the OLs. When we talk about ribosomes, they have two subunits, okay? We're going to say this subunit up here is bigger than this one down here, right? So it's pretty straightforward. This is the large subunit or ribosomal subunit. And then this one down here is the small ribosomal subunit, okay? Now, these have different ways that we can kind of like describe their size, okay, large and small, according to a Svedberg unit. And eukaryotic cells, that Svedberg unit for large ribosomal subunits, are called 60S, large ribosomal subunits. And the small and eukaryotic cells are called 40S, ribosomal subunits. But we sometimes, generally, in textbooks, refer to them as 80S ribosomes in eukaryotic cells. You're probably like, Zach, those numbers do not make any sense. 60 plus 40 is 100, Zach. Are you losing your brain? I promise you that the way that they do this via the Svedberg unit gives you an 80S ribosomal subunit for eukaryotic cells. In prokaryotic cells, it's the same concept. Again, we're not going to write these down, but this is your large. Here we'll put large ribosomal subunit, small ribosomal subunit. And cool, I'm glad that I know that now. Why do I need to know that? Before we talk about why I need to know that, the next thing I need you guys to remember is what are ribosomes made up of? You guys need to remember this. Ribosomes contain a very specific kind of molecule, if you will, that's kind of sitting in a part of it, very integral to its structure. What is this? It's got little like nucleotides on it. It's RNA. So ribosomes contain two different types of things that make them up. It's equal to RNA and what else? Proteins. So proteins. So we're talking about, remember when I said in the beginning, translation requires three types of RNA? mRNA, tRNA, and RNA. We usually just say ribosomes, but ribosomes contain RNA and proteins. Now, why did I spend the time talking about all this stuff? A common clinical relevance here is that they love to say, when you're talking about prokaryotic cells, prokaryotic.
Imagine being able to build anything just by thinking about it. Meet base 44.
Cells, okay? We can use different types of antibiotics to target these ribosomal subunits in prokaryotic cells. For example, if I give someone an antibiotic like an aminoglycoside, and there's so many different types of these, but the commonly one that you need to know is like gentamicin, and another one called tetracyclines, and there's a bunch of different types of these, doxycycline, tetracycline, minocycline, all those. Prokaryotic cells, it's technically not methionine. It's called informal methionine. So what is it called? Informal methionine. Okay, we'll put met. So again, the start codon is AUG in prokaryotic cells, same as it is in eukaryotic cells. But what it codes for is not methionine like it is in eukaryotic cells. It's called informal methionine. Sometimes it's even abbreviated as FMET. Okay? Either way, that's my start codon. So we're going to put here AUG. On this mRNA, there is a sequence of nucleotides, particularly like purines, that are a couple nucleotide bases upstream towards the 5' end from that start codon. And for whatever reason, they love to give this a particular name because this is where your ribosomes, a lot of initiation factors, things like that bind and recognize the mRNA in the prokaryotic cells and bind and helps to start the translation process. And this sequence that's like 8 nucleotides upstream from the AUG is called the Shine-Delgarno sequence, okay? And if you really want to know, it contains a lot of A's, adenines, and guanines, okay? So it contains a lot of adenine and guanines, or your purine nucleotides in that region, okay? So there's a Shine-Delgarno sequence. It's kind of like an identifier on the mRNA. And what happens is, A couple things. First thing is you have your small ribosomal subunit, okay? Your small ribosomal subunit will come and bind to this area, right? And what happens is when it binds to the area here on the mRNA, it uses a very special type of protein. Let's represent these in brown. Actually, no, let's do it in pink, so it's kind of different here. There's these things called initiation factors. And there's these initiation factors that recognize the Shine-Dalgarno sequence that are in the small ribosomal subunit, or bound to the small ribosomal subunit. And so what happens is the initiation factors in the small ribosomal subunit will bind the Shine-Dalgarno sequence. Then once it does that, it starts kind of moving towards the start codon. So two things happen. These pink things called initiation factors that are associated with the small ribosomal subunit will identify the Shine-Dalgarno sequence. When they bind, they then move down about 8 nucleotides until they hit the start codon, which is AUG. That's the first thing, okay? So if we wanted to kind of show that, that's the first event to happen. Let's put 1 here. First event to happen is initiation factors and small ribosomal subunits bind Shine-Dalgarno, move down until they hit the AUG. The second thing to happen here is that there's a molecule called tRNA, right? And tRNA is going to have anticodon specific to this AUG, which is UAC. And it'll be carrying with it an amino acid. What is that amino acid? Specific. We already kind of talked about it. We're going to abbreviate it called FMET. Now, when the tRNA comes, what is this called? This is your tRNA containing the FMET. When it comes in, as its anticodons interact with the codons here, there's something that helps to bring it or drag it into this area. What do you think that is? Let's represent another little pink color. There's a pink protein that kind of helps to yank that tRNA, the initiated tRNA, right, which contains the FMET, and bring it into where the start codon is. What is that pink protein called? It's called an initiation factor. That's it. So, first step, initiation factor, small ribosomal subunit, bind Shine-Dalgarno, move down until they hit the start codon. Second step, initiator tRNA and the FMET. When it comes in, as its anticodons interact with the codons here, there's something that helps to bring it or drag it into this area. What do you think that is? Let's represent another little pink color. There's a pink protein that kind of helps to yank that tRNA, the initiated tRNA, right, which contains the FMET, and bring it into where the start codon is. What is that pink protein called? It's called an initiation factor. That's it. So, first step, initiation factor, small ribosomal subunit, bind Shine-Dalgarno, move down until they hit the start codon. Second step, initiator tRNA in the prokaryotes, which contains tRNA and formylmethionine, with a initiation factor, come to the area where the start codon is and bind. That's the second step. Third step, there is a molecule bound to this initiation factor. And that molecule is called, let's bring it over here, a GTP. This GTP is a high energy molecule. What's going to happen is this initiation factor will break down the GTP into GDP and an inorganic phosphate. That will create a lot of energy. And what happens is at the same time the GTP gets broken down into GDP and inorganic phosphate, the large ribosomal subunit will represent it like this. The large ribosomal subunit will come over and bind into this area. And so what will it look like if we had kind of like showing all this happening here? This process and the large ribosomal subunit coming in here. This would be in your third step. So third step here is GTP gets broken down to GDP and inorganic phosphate, and the large ribosomal subunit comes and gets added in. What would be the final thing that would look like if we drew it down here? If we drew it all down here at the end product here, you would have what? Large ribosomal subunit, small ribosomal subunit bound here. Then what else would we have? We would have the tRNA kind of sitting in here with the F in formylmethionine bound with the codon. In this case, it would be AUG. And then what would we have released during this process? We would have released GDP and inorganic phosphate. And what else would we release? We don't need this thing anymore. We don't need this pink protein anymore. The initiation factors. We can just spit those out as well. So we can spit out the initiation factors as well. What are these things called? We're just going to abbreviate them initiation factors. So to recap really quickly, I know it's a lot of crap in one thing. Shine-Dalgarno sequence, identifier of the mRNA. Small ribosomal subunits, initiation factors, bind to it, identify it, move down to the hit the start. Second thing, tRNA, which contains the FMET, right, which is particularly based upon the anticodons complementary to the codons in mRNA. It gets brought to this area by the initiation factors. They bring it to the area and bind the tRNA. Then, third step, there's a GTP associated with the initiation factors. It gets broken down into GDP and inorganic phosphate. At the same time, a large ribosomal subunit will bind, and what will you get at that process? You get the large and small bound to the mRNA with the tRNA sitting in the ribosome in what site? We didn't talk about this yet, but there's 3 sites in a ribosome. One of them, if we start them here, this first one's called the A site. That's the kind of the arrival site. This one's called the P site, and this one's called the E site. And we'll go through these all in detail, but that tRNA is going to be sitting right smack dab in the middle, which is going to be the P site. Okay? So that covers the initiation and prokaryotic cells. Thank goodness, in eukaryotic cells, it's pretty much the same. We just give different names for stuff. So this step here in initiation, this is for particularly what? Eukaryotic cells. Subunit and other initiation factors that we're not too concerned with just yet that'll come in, interact with this mRNA. So let's draw here your small ribosomal subunit. That'll come and bind. That's the second thing that'll happen. And then what else is happening? You're having some initiation factors, some subunit. An initiation factor will come and bind to this 5' end. And we call this a eukaryotic initiation factor type 4. It'll bind to this 5' end. Okay? That's the first thing that'll happen. Subunit, that'll come and bind. That's the second thing that'll happen. And then what else is happening? You're having some initiation factors, some small little initiation factors that'll help that small ribosomal subunit to bind to the mRNA. This is the third thing that happens. Okay, so far we've had 2 things happen. Eukaryotic initiation factor type 4 identifies the mRNA. Second thing is the small ribosomal subunit with the initiation factors bind to the mRNA. The third thing to happen is that you have a eukaryotic initiation factor type 2. Eukaryotic initiation factor eukaryotic cells. What is the start codon in eukaryotic cells? It's the same one we talked about prokaryotes, right? AUG. What's the difference? AUG and eukaryotes code for methionine. Is that you have AG, which contains the E site, P site, A site, will come and have its factors. Eukaryotic initiation factor type 2. They just wanted to be annoying. The next step, which is called elongation. So what's the next step that we're going to talk about here? The next step is probably one of the more difficult ones to kind of visualize, but this is called elongation. This is the second phase in translation. So let's pick up where we left off. We initiated the translation process. Let's pretend this is the same. Thank goodness. This is the same in eukaryotic cells and prokaryotic cells, but we're going to use a lot of the examples here in eukaryotic cells. So this is primarily going to be used in eukaryotic cells that we're going to be using this as an example. And it's because we're going to be using particular types of factors. Okay, so in this example, just so you know, it's the same in prokaryotes and eukaryotes. Just in this example, I'm going over it in eukaryotes. So let's pick up where we use specific factors. You'll see what I mean. So to see if you guys remember everything we just talked about up here. You had to initiate it, right? Small ribosomal, large ribosomal, have to bind. Initiation factors help that process. Break down GTP into inorganic phosphate and bring a tRNA, which contains a amino acid, the initiator, TR, finish the initiation in that order, okay? Because that's the order we're gonna have things coming in. Isoleucine as an example over there. Let's bring them back. Let's put here AUA as the next codon that I'm going to read. If that's the case, then what do I need to bring into this area? A tRNA. In order for me to bring, but just in this example, we're going to put codon specific to that. Let's draw that and bring him in here. So we're going to have him come into this step here, amino acid. And that amino acid in this example doesn't really matter, but it's called isoleucine, since we talked about that one before, factor type 1.
¿Aún no sabes cómo utilizar la inteligencia artificial en tu trabajo?
Eukaryotic elongation factor type 1 will bind this tRNA, which is going to have anticodons complementary to these codons on the mRNA in the A site. Now, once that happens, let's show what that would look like. So here, we're still going to have that same initiator tRNA right here, right? Which contains the methionine. And if you really wanted to know it here, this would be UA. It's going to have tRNA, which is UA. Gets added in, what do I spit out? I spit out GDP in an inorganic phosphate, and what else do I spit out? My eukaryotic along for it. And then we're going to show it in a zoomed in way, because it is important that you understand this. What happens is, there's a little kind of like nitrogen on this amino acid here. And what was this one, if you really want to remember? Isoleucine. That nitrogen comes over and attacks the carbon end on this amino acid that's in the P site. And you know those like little things when you were a kid? They're like little sticky things with hands on the end of it, and you can throw it, it can stick to something and kind of like suck it back in. That's kind of what this guy is doing. It's grabbing the amino acid in the P site and sucking it back onto it in the A site. And then what would it look like if we kind of did that process? So let's say that we did this process here. What would that look like? If this were to occur, I mean, see, and this is going to be helping to perform this process, taking and catalyzing it. Sucks it back into where that area is, like a little, kind of like hands, the sticky hands that yank it back in. In order for this process to occur, the ribosome has an enzyme kind of intrinsically associated with it. And this enzyme is called a peptidyl that we just talked about, is catalyzed by an enzyme intrinsic to the ribosome RNA and would have it's kind of like moving things along, moving whatever was in the P site into the E site. Moving what was in the A site, we need G, which contains the isoleucine and methionine. We want to have an A. We spit out here. Eventually, you hit a stop codon, okay? And let's say that we use any of the three stop codons. Do you guys remember the memory trick? You go away. You are OTRNA with an amino acid will be brought into this step. Instead, what am I going to bring in? You all go away. You are gone. In that A site, am I going to have a tRNA come in and interact? No. No tRNA will be coming into this step, sir. So no tRNA with an amino acid will be brought into this step. Instead, what am I going to bring in? I'm going to bring in something called a release factor. So I'm going to bring in something called a release factor. A release factor has like a little pocket, if you will, that'll come in and interact with that UAG, that stop codon. It'll then prevent the ribosome from continuing to move along the mRNA, continuing to translate it. So it'll bind to the stop codon, stop the translation process, and then what? Shing! Cleave, shiatsu, that peptide away from the tRNA that's in the P site. So what else will it do? That's three things. What I want you to remember. Binds the stop codon. Second thing is it stops translation. Third thing is it cuts peptide in P site. So then from here, that release factor will then use its little shiatsu and cut that bond right there, separating the tRNA from the peptide. And then we'll have this peptide will then get released. And then from there, once we've released this peptide, it can go and do whatever it needs to do. Maybe it's going to get incorporated into the cell membrane. Ribosomes. We talked about the genetic code. We went through the phases of translation. And we talked about particularly how translation is occurring on ribosomes, right? With the mRNA, the tRNA. We talked about all that stuff. But here's the thing. Translation or protein synthesis can occur on ribosomes that are just kind of like freely circulating in our cytosol or cytoplasm, or it can occur on membrane-bound ribosomes, which are bound to what's called the rough endoplasmic reticulum. And you guys should be asking, when do I do it on the rough ER? When do I do it in the cytoplasm? And what occur on the rough ER and not in the free ribosomes is secreting the protein, embedding it into the membrane, and becoming a part of lysosomes. So now let's talk about the difference between the translation process that's occurring on a free ribosome and when it has to bind or translocate from that. And you make a very specific sequence of and the signal sequence is bound to the signal recognition particle, which is bound to the signal recognition protein receptor. You keep taking that ribosome. Here, we'll just draw the ribosome. The ribosome is going to kind of really line up very briefly. All right, guys. So at this point in time, we have gone from